Okay, all of you, chalo. So we have now the second example. Now we will find this very easy. The following results are obtained in several dihybrid test crosses in an organism considering three gene loci, capital A small a, capital B small b, capital B small c. Mind you, these are dihybrid test crosses and between three gene loci, capital A small a, capital B small b, capital C small c. And in a test cross TC with capital A small a, capital B small b, the number of recombinants are C and the parental combinations PC are 50 and 950 respectively. In test cross with capital A small a, capital C small c, the number of recombinants and the number of parental combinations are 75 and 425 respectively. And in a test cross with capital B small b, capital C small c, the number of recombinants are C and parental combination PC are 160 and 640 respectively. So the question is, based on the above result, identify the gene loci of the three genes or prepare a genetic map of the three genes. So depending on these, we have to find out कि chromosome के ऊपर where is A gene, where is B gene and where is C gene. Let's use this beautiful question this is. So solution. For capital A small a, capital B small b, number of recombinants are 50. Number of recombinants are 50 for capital A small a, capital B small b. The number of parental combinations are 950. Number of parental combinations are 950. So, how many map units or percentage or centimorgans it is? You know the formula now. Total number of recombinants upon total number of progeny or offspring multiplied by 100, which is recombinant कितने हैं 50, total number of progeny कितने हैं 950 plus 50, that is 1000 multiplied by 100, which is 5% recombination or map units or centimorgans, and that means the distance between A gene and B gene is 5 map units or percentage recombination or centimorgan. Next, for capital A small a, capital B small b, number of recombinants are 50. Number of recombinants are 50. I'm sorry. For capital B small b, capital C small c, I'm sorry. Number of recombinants are 160. Number of recombinants are 160 and parental combinations are 640. Parental combinations are 640. Thus, what is the COV crossover value? Total number of recombinants upon number of progeny offspring into 100. How many recombinants do we have? 160. How many total number of offspring do you have? 160 plus 640 multiplied by 1000, that means 160 upon this multiplied by 1000 is 20% recombination or map units or centimorgan, which means the distance between capital B and capital C genes, capital B and capital C genes is 20 map units or percentage recombination. How much was it for capital A small, capital A, capital B? For capital A, capital B, it was 5 map units. For capital B, capital C, it is 20 map units. Similarly, for just a sec. Similarly, for capital A, small a, so we do it for all three. We do it for all three, and what do we eventually get? We get that the distance between A and B gene is 5 map units. The distance between A and C is 15 map units. and the distance between B and C genes is 20 map units, or percentage recombination, or centimorgan. So now check that. So there are three possible combinations of the arrangement of A, B and C genes. Let's see which are these three combinations. You may have gene A, then B, then C. Or you may have gene A, then C, then B. Or you may have gene C, then A, then B. Now, how much is the distance between A and B genes? 5 map units, or percentage recombination, or centimorgan. How much is it between A and C genes? 15 map units. How much is it between B and C genes? 20 map units. समझो it is ABC. Now, A और B के बीच में कितना डिस्टेंस है? A और B का डिस्टेंस है 5 map units. B और C का कितना डिस्टेंस है? B और C का डिस्टेंस है 20 map units. So, how much is the total distance? 25 centimorgan of map units of percentage recombination. But distance between A and C, the two ends of the three G's, A and C, the two ends is 15 map units of centimorgan, and 25 sum does not match the 15 ends. 25 and 15 don't match. ये है 25 और ये है 25 और ये है 15, तो ये मैच नहीं होता है, therefore it is a wrong combination. Now let's see the other arrangement. Let's see the other arrangement. The distance of ACB, पहला वाला था ABC, वो wrong है. Now see ACB. ACB A और C के बीच में, A और C के बीच में कितना डिस्टेंस है? A और C के बीच में 15 map units. A और C के बीच में 15 map units. C और D के बीच में कितना डिस्टेंस है? C और D के बीच में डिस्टेंस है 20 map units. And total is how much? 35 centimorgans or map units or percentage recombination. But the distance between AB, the two ends of the chromosome, is how much? 5 map units. A और B के बीच में कितना है? 5 map units or percentage recombination or centimorgan. 35 does not match 5 ends of the sequence. Therefore, this is also a wrong arrangement. So, ABC is wrong and ACB is wrong. Now let's see the last combination, CAB. Now, C और A के बीच में कितना डिस्टेंस है? C और A के बीच में 15. C और A के बीच में कितना डिस्टेंस है? 15 map units of percentage recombination और centimorgan. A और B के बीच में कितना डिस्टेंस है? A और B के बीच में डिस्टेंस है 5 map units. 5 map units. 20 centimorgans of map units of percentage recombination. And the distance between CB, the two ends of the 3 gene sequence, is how much? CB का distance कितना है? 20 map units. 20 map units. Hence 20, the sum of A and B, AC or AB, is 15 plus 5, 20. And the CB का distance है 20. So 20 equal to 20 centimorgan or map units or percentage recombination. Hence CAB is the correct arrangement of genes on the chromosome. चलो, तो ये आपको nicely समझ में आ गया है. All you do is first use the logic of recombinant, parental combination, use COV, crossover value, total number of recombinant, total number of progeny offspring multiplied by 100, get percentage recombination or map units or centimorgan, and do it for AB, do it for BC, and do it for AC. and all three percentage recombinations you get it or map units or centimorgan, and then you find out which sequence is matching. So in this case, ABC is not matching. In this case, ACB is not matching. But in this case, CAB is matching. I hope you all have understood this now. Good. Sure. So we now go into the next question. Percentage of recombination between A and B is 9%. A and C is 17%. B and C is 26%. Then the arrangement of genes on the chromosome is. Now this will be very simple. We have three options: ABC, ACB, BCA and TAC. ABC, ACB, BCA, TAC. तो इनमें से कौन सा arrangement of genes is correct? Now you will find it very simple. It should be very easy now. Percentage of recombination between A and B is 9%, which is same as 9 centimorgan or 9 map units. Percentage of recombination between A and C is 17% or 17 centimorgan or map units. Percentage of recombination between B and C is 26%, that is 26 centimorgan or map units. Hence, BA के बीच में कितना है 9 मैप यूनिट्स. BA के बीच में कितना है 9 मैप यूनिट्स. AC के बीच में कितना है, AC के बीच में कितना है, AC के बीच में है 17 मैप यूनिट्स. So this is 9, this is 17 and BC का कितना है, BC का है 26 मैप यूनिट्स. BC का कितना है 26 मैप यूनिट्स. So 9 plus 17 equals 26. So what is the correct sequence? B, A, C. The correct sequence is B, A, C. And now this becomes very simple. I'm sure you all have understood this. Check. Now let's solve the examples from the NTA paper one by one. जो आपको NTA में है वो वाले solve करते हैं. Individual homozygous for small x small y were crossed with Y type plus plus, which you know means dominant gene capital X capital Y. Small x small y plus plus का मतलब क्या हुआ? Capital X capital Y. F1 hybrid produced was test cross. It produces progeny in the following proportion: plus plus, which is capital X capital Y 900 parental. Small x small y 880 parental plus y, in other words capital X small y 115 recombinant plus x, in other words capital Y small x 105 recombinant. Find out the recombination frequency. Find out the recombination frequency, and we know it now. Number of recombinants are how many? Plus y is 115. Plus y 115. Plus x is 105. Plus x is 105. That means recombinants are 220. And total number of progenitor offspring is 900 plus plus. 900 plus plus parental. 880 XY, 880 XY, sorry, 880 XY is parental. तो XY parental कितने हैं? XY parental are 880 and the plus plus capital X capital Y parental are how many? 900. And how much are recombinant? 115, 105. 115, 105, 220. And what is the total number of offsprings? 900 plus 880 plus 115 plus 105, a total of 2000. So, what is the percentage recombination, also called as crossover value? total number of recombinants upon total number of progeny offerings into 100, which is 220 upon 2000 as calculated above into 100, which is 11% recombination, which is 11 map units or 11 centimorgans. So this is very simple. We have understood this well. ये NTA में आया था, इसको याद रखना. Example 2. An individual homozygous for gene small d is crossed with wild type and F1 is crossed back with double recessive. Appearance of offspring is as follows: plus that is parental 903, small d parental 897, plus small d recombinant 98, C plus recombinant 102. Question: the distance between genes C and D is 20 map units, 9.8 map unit, 10.2 map unit, 10 map unit. This is again from NTA paper. Now it's very simple for you. Number of recombinants are plus D. Plus D is how much? 98. Plus C is how much? 102. That is 200. And total number of progeny offsprings is how much? 903 plus parental 897 CD that is parental and wild type is plus. So how much is wild type? Parental 903. How much is CD? Parental 897. And how many are recombinant? 98 and 102. तो याद रखना हमेशा जब भी प्लस एंड छोटा अल्फाबेट है, प्लस एंड छोटा अल्फाबेट है, व्हाट इज द मीनिंग ऑफ दैट? इट इज रिकॉम्बिनेंट. और जब छोटा अल्फाबेट है मींस इट इज पेरेंटल, या प्लस है इट इज पेरेंटल. Simple to remember, very easy to solve. Plus means parental, small is parental, plus small is recombinant, plus small is recombinant. So simple now. And what will be total number of progeny offspring? Parental 903, parental 897, recombinant 98. Recombinant 105, a total of 2000. So what is crossover value? Total number of recombinant upon total number of progeny offerings into 100 is 200 upon 2000 into 100. That is 10% recombination or 10 map units or 10 centimorgans. Simple. This is in the NTA paper. I'm sure you're finding this very easy now. Very good. And then you are having another example from NTA. ये अच्छा क्वेश्चन है. In a crop of Drosophila, heterozygous member with grey body, small b plus and long wings, VG plus. तो नाम से मालूम पड़ रहा है this is heterozygous, was crossed with black body, small b and vestigial wing VG. So what is the meaning of that? This is a test cross. समझ आ रहा है? This is a test cross. Small b small plus and VG plus is heterozygous F1 ratio. Crossed with black body and vestigial wing VG is a test cross. The progeny has the following ratio: grey vestigial 24, grey long 126, black long 26, black vestigial 124. What is the frequency of recombinants in the population? And the four options are given. Next question. Please understand this. What are the number of recombinants here? Number of recombinants are, ध्यान से, plus VG. Plus VG is a recombinant. plus Vg, gray and vestigial, as I said, plus and chota alphabet is the recombinant. So 24 is plus Vg and small b plus, again small alphabet and plus is a recombinant. So 24 plus 26 equals 50. What is the total number of progenitor offsprings? Total number of progenitor offsprings are plus plus parental 126. BG small 124 parental plus VG 24 plus and chota 24 recombinant B plus small alphabet plus 26 recombinant, and that means total number of offsprings are 126 parental, 124 parental, 24 recombinant, 26 recombinant, that is 300. How much is number of recombinants upon total number of offsprings multiplied by 100 COV? That will be 50 upon 300 multiplied by 100 is 16.7% recombination or 16.7 map unit or 16.7 centimorgans. Simple. Again from the NTA, now we find it very simple. Another example. ये अच्छा क्वेश्चन है. Two linked genes small a small b show 20% recombination. Small a small b, how much is the recombination? 20%. The individuals of a dihybrid cross between plus 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 crossed with small a d shall show gametes, and four options are given: plus plus 80, AB 20, plus plus 50, AB 50, plus plus 40, AB 40, plus A 10, plus B 10, plus plus 30, AB 30, plus A 20, plus B 20. तो ये चार में से what will be the individuals of a dihybrid cross between plus 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 small a small b, and what will be the gametes formed if A and B are having 20% recombination? Good question. Individuals of a dihybrid cross between plus 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 crossed with small a d will show what gametes? ये ध्यान से सुनना. Plus 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 small a d are parents. So what will be the gametes formed by them? Plus plus slash plus plus and small a small b. So what will be the gametes produced from them now? This is double heterozygous. This is double heterozygous. Will form gametes which are homozygous. Which are single heterozygous. Double homozygous, double. तो उसमें से जब gametes बनेंगे, what will they become? Instead of 4, 1, 2, 3, 4 G, there are 2 G. Instead of 4 G, there are 2 G. So, what has the plant or the parent become? Gametes. What will be the combination of the gametes possible? Combination of gametes possible will be plus plus में से एक plus जाएगा and plus plus का एक plus जाएगा. This is slash. So plus and plus and check this here, plus and small a and plus and small b plus and small b, and small a and small b. So, how many types of gametes are possible? All four types of gametes will be formed. That is, plus is formed, plus is formed, plus a is formed, plus b is formed, and ab is formed. चारों टाइप के गैमेट पॉसिबल है. So options 1 and 2 above are incorrect. Options 1 and 2 of the question are incorrect. Options 1 and 2 of the question are incorrect because they are showing only two types of gametes. They are showing only two types of gametes. तो ये दोनों incorrect है. And how many types of gametes are possible? All four. So 1 and 2 are not possible. So now let's see which are possible. Option 4. Option 4 plus 30. AB 30 plus A 20 plus B 20. Fourth option of the example. What are the number of recombinants? Recombinants are plus A 20. Plus A कितना है? 20. And plus B कितना है? 20. So total कितना हो गया? 40. And what is the total number of offspring? Plus is 30. Parental. AB is 30 parental. 20 are plus A recombinant. 20 are plus B recombinant. So total is how much? 100. So crossover value will be total number of recombinants upon total number of progeny offspring into 100 will be 40 upon 100, which is multiplied by 100. 40 upon 100 into 100 is 40% recombination or 40 map units or 40 centimorgans. In the question it is given two linked genes small a and small b show 20% recombination. If you remember, what are the examples? Two linked genes small a small b show 20% recombination. And what does the fourth option show? Fourth option shows there is 40% recombination. So option 4 shows 40% recombination is wrong. ये wrong है. And now let's see the third option. Third option कितना आएगा? Third option आएगा plus is 40%. And AB is 40 plus A is 10 plus B is 10. And check this out. Plus is 40, AB is 40 plus A is 10 plus B is 10. How much is plus A recombinant? 10. How much is plus B recombinant? 10. So what are the total number of recombinants? 20. What are the total number of progeny offspring? 40 plus parental. 40 small b parental plus A 10 recombinant plus B 10 recombinant, a total of 100. So crossover value, number of recombinants over number of progeny offerings into 100 is 20 upon 100 into 100, which is 20% recombination or 20 map unit or 20 centimorgan. In the question it is given two linked genes small a small b show 20% recombination. And what has your third option shown? Your third option has shown that this combination is 20% recombination. That means option number 3 is correct. It is 20% the distance between small a small b the other gene small a small b showing 20% recombination, उनके gametes कौन से बनेंगे? उनके gametes बनेंगे 30%. उनके gametes कौन से बनेंगे? This option. उनके gametes बनेंगे plus 40 small a small b 40 plus a 10 plus b 10. Showing 20% recombination between gene A and gene T. बहुत अच्छा क्वेश्चन था. I hope you all have understood this well. So what did we do in this question? By forming gametes, we know there are 4 types of gametes and 4 types के gametes बन रहे हैं. So option 1 and 2 are wrong. ये gametes बन ही नहीं सकते हैं क्योंकि इसमें सिर्फ दो गैमेट्स हैं, 4 टाइप के गैमेट नहीं है. Then we did ruling of third and fourth and उसमें चारों गैमेट्स हैं. But what was in the percentage recombination of fourth option 40% and A और B के बीच में कितना है 20%, तो वो wrong हो गया. And option 3, what was the percentage recombination of this option? 20%. So that is 20 map units. So if a 2-linked gene small a small b showed 20% recombination, option 3 is 20% the correct option. So please refer to your book and pause the video for a second and you can check this question out. Then you continue with the next numerical. Example 5. What will be the linkage strength between two genes capital A, capital B, which are 6 centimorgan apart from each other on a chromosome? 6%, less or 50%, 94%, data insufficient. Check this out. Two genes capital A, capital B are 6 centimorgans apart on a chromosome. Now, ध्यान से 6 centimorgan is 6% recombination or 6 map units. We know it very well now. How much is 1% recombination, 1 map unit or 1 centimorgan? So how much is 6 centimorgan? 6% recombination or 6 map unit, which means the linkage strength is very high and the distance between genes A and genes B is very less. A और B के बीच में सिर्फ 6% recombination possible है, which means these are tightly linked genes. They are tightly linked genes. And the linkage strength कितना होगा between two genes A and B? 100% minus 6%, which is 94%. So how much is the linkage strength? 100% minus 6%. सेंटीमॉर्गन को पहले हमने किसमें कन्वर्ट किया? सेंटीमॉर्गन को हमने पहले कन्वर्ट किया परसेंटेज रिकॉम्बिनेशन में. So 6 centimorgan is 6% recombination. And how much is the distance between two genes A and B? 100% minus 6%, that is 94%. So, next question, I'm sure you all have understood this. We'll just take a small break again and I will come with example 6.